Hi everyone, it's a pleasure to meet you, and I'm so honored to receive this award. I am Agata Lemiesz Patriotis, a Postdoctoral Fellow at the Koch Institute at MIT in the lab of thylacic JAKs. I have always been fascinated by how cells make decisions, how they choose what to become, and how those choices can wrong in cancer. That curiosity led me into epigenetics, the study of how DNA packaging and chromatin structure control gene activity beyond the genetic code itself. What excites me about this field is that it adds a new layer to our understanding of disease. Even when the genetic code is the same, the way it's read, the way it's interpreted, can completely change a cell's behavior. And that's where many answers in cancer biology lie. My scientific path started in Poland, where I was born, continued through a Fulbright Fellowship at the University of Virginia, and then to the Rockefeller University, where I had the privilege of training with David Allis, who pioneered modern chromatin biology. Now at MIT, I focus on pancreatic cancer, one of the most aggressive human cancers, and one where, despite decades of genetic discovery, patient outcomes have barely improved. My research asks, how do pancreatic tumors change their identity to escape treatment, and can we reverse that? These tumors depend on a mutation called KRAS. When KRAS is blocked, instead of dying, the cells often reprogram themselves, becoming more primitive, mobile, and resistant to therapy. I study the epigenetic regulators that enable this switch, the chromatin-remodelers and histone modifiers that normally keep the genome organized and cell identity stable. When these regulators malfunction, as they often do in pancreatic cancer, cells lose their sense of identity, become invasive, and adapt to survive. My goal is to understand this process and develop what is called differentiation therapy. Not to destroy every cancer cell, but to push them back towards a stable, less aggressive state. If we can lock tumors into one identity, we could make existing treatments far more effective, not just in pancreatic cancer, but also in other cancers that share this plasticity. In the long term, I hope this work helps bridge basic chromatin biology with patient-centered research, taking what we learn about the epigenome in the lab and translating it into new therapeutic strategies for people facing these devastating diseases. This award gives me the freedom and visibility to take the next step toward translating these findings into something clinically meaningful. I am deeply grateful to the Hopper Belmont Foundation for supporting early career scientists and for recognizing the importance of curiosity-driven research. Thank you so much. I'm truly honored to be a part of this year's Inspiration Award group.